Okay, so what am I going to talk about? I'm going to talk about pangenomics and machine learning for crop improvement. My name is Dave Edwards. I'm the director of the Centre for Applied Bioinformatics here at the University of Western Australia. And here is my contacts. The slides will be shared afterwards. And I'd be very happy for anyone to contact me because we thrive on collaborations. A little bit about me. I did my first degree in agricultural science at the University of Nottingham, which is something I've just learned I share with Anna, which is interesting. After that, I worked for a company called ICI Seeds, which eventually became Syngenta. I traveled around the world, hitchhiking around, got a job at the University of Adelaide, University of McGill, and eventually came back to the UK and did a PhD in plant sciences at the University of Cambridge. Since then, I've traveled around quite a bit in different jobs, but currently I lead an applied bioinformatics group here at the University of Western Australia. I work on genomics, diversity, and machine learning. We work with lots of different species. So much of the early work has been on brassicas, a lot of work on wheat, chickpea, pigeon pea, soybean, lupin, clover, pea, lentil, and quite a few others, quite a few minor crops as well. We also work on data sets of just general interest. So Hakia, most of the people in the audience wouldn't know what this is. It's native to Western Australia, and it's adapted to very low phosphorus conditions, to the point that if you planted it in normal phosphorus soil, it dies of phosphorus poisoning. We work on seagrasses from the evolutionary perspective, same with amberella, sister species to all flowering plants. We've done a little bit of work on humans, one of the most boring species I've come across in terms of the genomics. We've worked on chicken, fish, coral, and lots of other bizarre things. I teach practical bioinformatics, and I established recently a Master of Bioinformatics here at the University of Western Australia. I also established and am the founding director of the UWA Centre for Applied Bioinformatics, which tries to bring together the biomedical and environmental and the agricultural bioinformatics across not just UWA, but across universities in Western Australia, and also linking out to organizations such as EMBL. And this has been running for about a year and a half now, and seems to be pretty successful with about 150 full-time members. We also support students in student projects in applied bioinformatics, and we host quite a few researchers in this space. So what drives me? Well, my first degree was in agriculture, and that is still at my heart of what I like to do. So agriculture is clearly the greatest success story. If you look at the growth of the human population through the ages, it's grown massively, and this has been supported by the ability to produce food on a scale that was inconceivable for thousands of years. Most of these changes have occurred through changes in agricultural production, and this illustrates how some of that has changed. So if you've got the picture on the top, the wheat crop is very tall, it's manual labor, it's mixed agriculture, and now you have modern agriculture where everything is seeded directly into rows and fully automated with optimized crops and inputs. And this has enabled massive increase in agricultural productivity, changes in society, and possibly fueled, and probably has fueled the population growth, which is causing a lot of the problems that we're seeing today. Why is this allowing me to do this? Oh, here we are. It's not all good news. The climate is changing. The growing population is causing climate change. That's clear now. We are polluting the environment with CO2 and lots of other chemicals which are not good for it. And this climate change is already starting to impact agriculture. It's very clear here in Western Australia, we are seeing changes in rainfall patterns and changing in cropping according to this. And it's especially in Western Australia, it's going to have a negative effect. In some regions of the world, it'll have a positive effect, having longer growing seasons. So what you're going to see is you're going to see a shift in overall agriculture as to where things are grown. What's most troubling is the most food insecure regions, especially around the equatorial regions, around Africa, are going to suffer the worst in terms of productivity, and they're already very food insecure already. So globally, this is a really bad issue. So we're facing an uncertain future. I mean, how do we maintain food security? Well, clearly there are social and economic and environmental factors we can play here. Certainly having wars in Europe doesn't help, but that's not my area. So I'm going to focus on what I do know about, and that is agriculture and the sort of continued improvements that we can have in agriculture. Now, many of the developments that we've had have been through things like improved use of fertilizers and pesticides and improved farming practices, as illustrated by the images I was showing. But there's a limitation as to how far you can go with these. There's only so much fertilizer and pesticide you can actually put on the ground, and there's quite a push to reduce the applications of farm inputs. Farming applications in developed countries are really quite advanced. They could have been improved in other regions, but again, there's likely to be limits that people will come up to in being able to improve agriculture through those practices. So one of the most promising areas for improving crop productivity is improving germplasm. And one of the routes for improving germplasm is through the use of genomics. So what we need are new crops for a new climate. It's clear that the major crops will continue to provide the majority of calories. And for these, we need to focus on yield improvement and adaptation as the climate changes. But there are many minor crops that are important for developing countries, things like pigeon pea that a lot of people don't really work on, but they are important for food security. These have got the greatest potential for improvement. And if we can improve productivity and sustainability and expand utilization of these, it could provide greater security through diversifying the sort of crops that people are eating. So if we're going to use genomics, we've got to understand what genomics is. And I'm pretty sure most of the people in the audience will understand genomics. For a little bit of a personal history, this image on the left, this is how I first started sequencing DNA. Some of you will see me, I've got quite a few white hairs, but I'm not that old. So this has been around for a while and was for a long time the main way to be able to sequence DNA. And you could get a few hundred bases a day from these sort of gels. It's really quite labor intensive. There's a reasonable amount of radioactivity involved. And this is the traditional Sanger sequencing method. This evolved through a whole range of different technologies and machines, and this is the ABI 3730 XL, which has been around for a long time now, and is still probably the best way to do Sanger sequencing that there is, using capillaries at the moment and fluorescent dyes. And this is still in common use. If you only want to sequence a short piece of DNA, this is the sort of machine that you would use. But things changed. Everyone's aware of the changes in next generation sequencing and how sequencing technology has come on. Back in, I think, 2006, 2007, my group bought the very first next generation sequencing machine in Australia, the 454 system. Looking back at that, the quality was terrible. The reads were quite short, but it was revolutionary at the time. And things have moved on, and it's incredible just how much you can get for relatively low cost at the moment. The Illumina sequencing still remains exceedingly high quality, exceedingly cheap for the amount of data that you get. But it does suffer from relatively short reads. Long read technology has come on leaps and bounds in the last few years, and what I've seen is a general competition between Oxford Nanopore and PacBio with the Sequel platform and the new Revio platform they've released. These technologies are always kind of outpacing each other. It's always difficult to know which one will be best to sequence a genome at any one time. I would say a year or two ago, I would have gone with Nanopore, and I did go with Nanopore. Currently, I'm favoring the PacBio Sequel. In 6 months' time, I don't know which one I prefer, but they're revolutionizing the way that you can sequence genomes. And you can sequence genomes telomere to telomere with these technologies relatively cheaply. And especially with the Illumina technology, you can go through and use sequencing, genotyping by sequencing methods to identify paths in pangenome graphs, identify SNPs, that kind of thing. So the sequencing technology, the main message, it's incredibly cheap. And you can sequence pretty much any genome now relatively easily. Some of my presentation goes into a bit of a historical work, so we need to put that into context of to what we could do at the time, and I'll do that then. So what can we do with all this information? Well, we can characterize genome variation. If I could actually see any of you, I'm pretty sure you all look very different, and many of those differences are genetic, they're heritable. We tend to look like our parents, our kids tend to look like us. And this is the heritable variation that's there. We can actually go through now and start sequencing vast numbers of individuals, vast populations, and start using that information to build the statistical models to be able to associate that variation in the genome with variation in traits. Much of this is really quite subtle, and so you do need relatively large numbers and quite large statistical models to be able to make those associations. When I first started working on genomics, there weren't many genomes, in fact, no genomes available. We were sequencing express sequence tags. Trying to identify candidate genes for traits was a real challenge. Now it's a lot easier because we can generate so much genomic information. We can start to make those associations and identify genes for traits. And we can start to apply molecular markers for breeding. I've been working with plant breeding companies for a long time. And in the early years, they wanted maybe a dozen markers at the most. They said any more than that was not necessary. They had their field trials. They knew what they were doing. Over time, as the cost of generating the markers reduced, and as the value of those markers increased and the technology started to change, then the breeders were asking for more and more markers. They were saying, well, a 3K array or a 6K array or a 60K array was really quite good. Until it got to the point where the breeders were saying, well, can we just go and sequence this population and find out what's there? And that's mostly been driven by the reduced cost of generating this data, but also breeders seeing the value of the genomic technology and the associations for breeding. And certainly, you wouldn't start a crop breeding project now without using molecular markers, without having a good understanding of the genomics and the genetics behind it. So we started sequencing genomes, and that was part of a whole range of different genome sequencing projects. If you sit down for dinner, if you look around the table, I'll probably sequence most of the things that are on the plate, all your brassicas and the and wheat and what have you. And we were in a situation where we were working closely with Bayer Crop Science, and we had quite a lot of funding to sequence some early genomes. And what we were seeing was when we were looking at these genomes, we were one of the first groups to actually have multiple genomes of the same species. And when we compared them, we saw that they were different. And this is early days of genome sequencing. Some of the technology wasn't really up there. And we knew that quite a lot of the variation that we were seeing was due to differences in the assembly quality, differences in the annotation quality. But when we looked quite deeply, we actually saw there was still a significant amount of variation that couldn't be accounted for by biotechnological variations. So what we were seeing was that genomes were actually different. And what I'm going to try to persuade you over the next few slides is that a single reference genome does not represent the diversity of a species. And if you're going to do association analysis in any species, you don't want to use a single reference. You really want to use a pangenome. I want to explain that presence-absence variation genes, so genes that are in some individuals but not in others, that they're responsible for important agronomic traits. And this is important for breeding and selection, but it's also very important for genome editing. So if you want to use genome editing as a method for crop improvement, you need to know the gene content, not just of the species, but of the individual that you're working with. So pangenome terms, well, there are quite a few different terms that are used in pangenomics, but the main ones are understanding what a core genome is. And a core genome is the DNA content that is shared amongst all the individuals of that species. So there's no real variation in these genes between individuals. Then you've got variable genes or dispensable genes. And these are in some individuals, but not in other individuals. And altogether, they make up the pangenome. And it's important to know that there's not a single individual will have the whole of the pangenome. A pangenome is shared across many, many individuals. So it's not representative of a single individual, it's representative of a species. So everyone has their own COVID story. Over in Western Australia, we were closed off from the rest of the world. They closed the borders. We could do what we liked within Western Australia, but we couldn't leave and no one else could come in. So I wasn't traveling, so I had to do something. And what I did, I made pangenomes and wrote pangenome papers for a whole range of different species. So that's what kept me busy during COVID. I'm publishing less now after COVID, bizarrely, because now I can actually go out and do other things. But this was what kept me busy. And the way that we build pangenomes is evolving. It's changing as the technology changes. It's changing as the DNA sequencing data changes. We focused initially on what we call the iterative assembly approach. We did this because at the time it was relatively expensive to generate sequence data. If you wanted to sequence a whole genome, you need to get very high coverage. If you wanted to do de novo assembly of multiple genomes and to compare them, That was just, it wasn't cost effective for most species you could think of. And again, you had many issues because these were short read assemblies, and then comparing the short read assemblies, many of the differences were not due to real biological differences, they were due to differences in assembly quality and difference in annotation quality. So we used the iterative assembly, which I'll describe in a bit. At the time when we were doing that, we looked at graph-based assemblies, and it wasn't possible to make graph-based assemblies with eukaryotic genomes. These we used in bacterial pangenomics. However, now the technology has changed. The quality of the data is so much better. The algorithms and the methods are so much better that the population graph is now the default way that we make pangenomes. And we actually combine population graphs with an iterative assembly approach to map populations, to map tracks through the graph for those individuals. Did I skip one there? No. So for the de novo assembly, we generate high coverage sequence data for each individual. We assemble each independently, and then we annotate each assembly, and we compare gene content. The issues are very similar. Genomes can have quite different annotations. So the annotation method, even just a one base difference can actually decide whether there's actually going to be a gene called there or not. And that causes problems when you're calling presence absence of genes. And differences in assembly or annotation, they can mask real differences in the pangenome. So this can be quite challenging, especially at the time when we started doing this, because the assembly qualities were relatively poor. The iterative assembly was quite useful because it didn't need a huge amount of data. We started with a reference. You've got to have a good reference to start with. You map the reads to the reference, and then you pool and assemble the reads that don't map. And these new contigs can then be added to the assembly. This can be iterative, so you can just keep on adding more data as you go through. It's important that you pool the number of individuals because often you'll have relatively low coverage data. So we're looking at least 10x coverage. 10x coverage isn't enough to assemble genes individually. But if you've got 100 individuals, maybe 20% of them contain a gene of interest, and you've got 10x coverage for those, you've got high coverage for that gene. So the maths kind of works out that it does allow you to assemble relatively rare genes as long as you have good pooling and diversity that's in there. So you can keep on repeating steps 2 and 3 with additional individuals. And eventually you get your reference that you started with and a whole heap of new contigs. You can start to play some of these new contigs where possible using paired end or mate pair data. This is all done with short read data. You can use linkage to equilibrium or genetic mappings in some cases to be able to place them. And then once you've actually got your pangenome, for each variety, you perform presence absence assessments on the reference. You remap all the reads, and then based on the mapping of the reads for each individual, you can call presence or absence of every single gene. So what you end up is a large matrix for all the genes and all the individuals, and presence absence of each of those genes across each of those individuals. And that was a founding point for a lot of our iterative assembly work early on. As I said, now we tend to use population graphs. Now, population graphs, they were pioneered in human populations where it's actually quite a bit easier to work because there's far less presence-absence variation in humans than there is in plants. These are now the foundations. We make population graphs for a range of different species, and even species where we made early iterative assembly pangenomes, we've now started using population graphs. They allow you to capture so much more information, relationships between different parts of the genome. So we make population graphs using as many high quality individuals as we can access or produce, and then use that graph to map data from a large number of individuals. And these can be hundreds, if not thousands of individuals mapped to the graph to be able to look at the variation across that population. So you don't need full whole genomes for all your population, just a subset to be able to build the graph. So I'll go through a bit of history. The Brassica oleracea pangenome was the first one that we did. And this came out from a project, a collaboration, an international collaboration many years ago. It was eventually published in 2016. But it was through, I think, the early hours of the morning in a hotel bar with many of these people at the Brassica meeting in Saskatoon, where we thought, well, what could we do to look at the diverse morphotypes on a genomic basis? At the time, it was still relatively costly to generate the sequence data. So individuals were coming up saying, well, I could provide 10x coverage of this individual, for example. And together with everyone providing at least 10x coverage for a range of different Brassica oleracea morphotypes, we could actually start to generate initially just some comparative analysis, but this would form the foundation of generating the Brassica oleracea pangenome. And this work was led by a PhD student of mine, Agnieszka, who's based in Gießen at the moment as a professorship there. I'm not sure if everybody knows about Brassica oleracea. Brassica oleracea have been called the dogs of the plant world because dogs all have the same genome, but they range in sizes and shapes and colors phenomenally. Brassicas are the same. Brassica oleracea, you have all these weird morphotypes. Broccoli looks very different to cabbage, looks different to cauliflower, kale and Brussels sprouts. They have a huge amount of diversity. So we were looking to see if we can identify some genomic basis for some of this diversity. So we made the Brassica oleracea pangenome. We did it using the iterative assembly approach. It was the first time we'd applied this. At the time, there were 2 genome assemblies of Brassica oleracea that were available. They had 54,000 and 45,000 genes each, mostly varied because the quality of the assembly and the annotation. Within the pangenome, and we were quite conservative in calling genes because we didn't want to call genes that we weren't sure were genes. So these are confident genes we're looking at. Over 61,000 genes in Brassica oleracea, and an increase in overall genome size as well. But one of the things that quite surprised us at the time was the extent of genes that were variable. 18.7% of the genes in Brassica oleracea that we're looking at were present in some individuals and not in others. And this is only looking at 10 individuals. When we tried to publish this, it was a bit of a challenge because the reviewers didn't believe it. The reviewers were quite critical and assumed that much of the variation we're seeing was due to technical errors. So we even had to go through and PCR up a whole range of these genes to show that they were missing in certain individuals. And we got 100% confirmation from the PCR, and that allowed the paper to go through and be published. So then we started asking questions of the data. So one question you can ask, we haven't sequenced that many individuals. How many more individuals do we need to sequence to capture as many Brassica oleracea genes as we think might be out there? So Agnieszka did this modeling where you've got an increase in the number of genomes that were put forward in different orders. And as you can see, the number of genes increases as you go through, but starts to level off. Similarly, the variable gene set, the core gene set tends to get smaller, but again tends to level off as you're seeing more genomes there. And if you run this through a mathematical model, you can predict how many genes are likely to be in Brassica oleracea if you just kept on sequencing thousands of genomes. And this number is very similar to the number of genes that we've actually identified. So we're pretty confident from this modeling that we've sequenced pretty much every Brassica oleracea gene that we could possibly find, just through sequencing just through analyzing the genome data for a relatively small number of individuals. So the next question we asked, well, these genes, they're present in some individuals, but absent in others. So are they useful? What's their value? Presumably, if the plant can live without it, maybe they're not that important. So we had a look at the annotations of these, the gene ontologies for the variable genes compared to the whole gene set. And what we saw that the variable genes were enriched in certain terms, and these were mostly around abiotic stress and biotic stress, especially disease resistance genes tended to show significant presence-absence variation. We had a bit of a hint a few years before this because we were searching for a few disease resistance genes in Brassica napus, and we knew where they should be. And we were seeing presence-absence variation there on a very small scale. To see it on a genome-wide scale was really quite phenomenal. Now, many pangenomes have been made since this Brassica oleracea pangenome, and almost all of them show very similar patterns of disease resistance-based genes and genes for abiotic stress showing presence-absence variation. One of the exceptions that I've seen personally is in the Amberella pangenome. And we think that may be associated with its evolution. It evolved on an island in New Caledonia, and it was separated from many other plants. So there's less likely of other diseases perhaps coming through because it was so isolated. And there we saw mostly abiotic stress resistance genes, abiotic related genes showing presence absence variation, and relatively few disease resistance genes. But when you look especially at crops, disease resistance genes are really up there. And when you look at disease resistance genes of different types, these are three main types of disease resistance genes in brassicas. If you look across the individuals that we analyze, you can see that cabbages have got far more disease resistance genes than cauliflowers, for example, and broccoli. So if you're looking to get some more disease resistance in your cauliflower or broccoli, or find novel disease resistant sources, then it would actually make sense to go out to cabbages and bring some of those in as potential sources of disease resistance. Now, Brassica oleracea is one thing, but the main thing that we were working on, the main cash crop, was Brassica napus. So we managed to get a few years later, get some more data on Brassica napus, many of which you will know as canola. We had resequenced data for 33 standard Brassica napus lines and 20 resynthesized Brassica napus lines. Now, these resynthesized lines are formed in the lab. Brassica napus is a polyploid. It contains the A genome from Brassica rapa, the C genome of Brassica oleracea, and they come together to form Brassica napus. This happened after domestication of the brassicas, so it's only a few thousand years ago. But the resynthesized were made recently, and these are essentially new Brassica napus where those genomes have been brought together to form new forms of canola. We use the Darmor reference as the standard. This is the best reference that was available at the time. We increased the pangenome assembly size, and we increased the number of genes significantly from that single reference in the pangenome. And when we looked at the variable genes, around 40% of the genes are variable between the different species, between the individuals. 60% were core genes. We showed that the variable genes were shorter and have fewer exons than core genes, and the number of variable genes indicates just how much diversity is present within these species. Now, one of the reasons we think there are more variable genes in Brassica napus compared to Brassica oleracea is because it's polyploid, and going through that polyploidy event, bringing the A and the C genomes together, it leads to redundancy of genes. So there's likely to be some genes lost in some individuals, but maybe maintained in others. Again, when we looked at the functions of these genes, plant hypersensitive response, many of them associated with disease resistance. At the time, we also were quite interested in the mechanism of presence-absence variation. So what causes genes to be lost, for example, or genes to be retained? And this, interesting, was one of the first times we applied machine learning into genomics. So what we did, we built a machine learning model using a whole range of different genomic-based inputs. For example, how close a variable gene is to certain types of repeat sequences, distance from centromeres, position on the chromosome, the gene length, features such as that. And then we built models for Brassica oleracea, Brassica rapa, and Brassica napus, because at the time we had a Brassica rapa pangenome as well. And if you remember, the Brassica oleracea and Brassica rapa are the diploids that form Brassica napus. And one of the things we found when we built an interpretable gene explainable AI model, where we could actually go through and ask what were the features that were leading the model to create its interpretation. We saw that the mechanism for gene loss varied between the diploids and the polyploids. And for example, the position on the chromosome was very significant in Brassica oleracea and Brassica rapa. But there are other features on Brassica napus, and particularly whether it's in a homologous block. And this had a major effect. And I'll explain that in, I think, the next slide. So as I said, the Brassica oleracea and Brassica rapa, they came together to form Brassica napus, but they originated from original founder Brassica, so the genomes are related. And A2 and C2 originated in an early form of Brassica before the divergence of rapa and oleracea. So they share a significant gene content, and they were brought together again for the first time in Brassica napus. And what can happen here is you can get exchange. I'll give you I'll first describe what this figure is. This is a chromosome going around, the chromosome A2 and chromosome C2. Each line, so all these green lines here is an individual. A red line is a synthetic individual, and where you have a dot, it's a missing gene. So what you can see, you've got some random genes that just seem to be showing presence-absence variation in some individuals. But you also see here, for example, a large region of a chromosome where you've got many lost genes. And when we looked into this in detail, what we saw was what's called a non-reciprocal homologous exchange, which basically means that the piece of DNA on the equivalent A2 chromosome was copied across to the C2, and it had 2 copies of the A2 version. But any genes that were then specific to C2, because there'd been quite a bit of divergence in that time, were then lost on that C2 region. This is important agronomically because we know that there's a flowering time gene that's in this region, and this individual has got a different flowering time due to the loss of that gene. So there are some agronomic aspects associated with this. One of the things we hadn't expected to see was in the synthetic lines. The synthetic lines had much more non-reciprocal homologous exchange, and not just going from the A genome to the C genome, but also from the C genome over to the A genome, but to a lesser extent. And this is important for breeders because breeders were generating these lines to get more diversity. But in fact, they were even getting more diversity than they were expecting because of this gene loss that was happening in these genomes. So that was quite valuable information. Next, we thought we'd give ourselves a challenge. So we made a bread wheat pangenome. We were fortunate enough to work with a collaborative group in Australia a few years before, where we managed to persuade the federal government to sequence a group of Australian lines, only to about 10x coverage. And this allowed us initially to look at diversity and look at SNPs. But we thought, well, this data is really quite useful for making an iterative assembly pangenome. So we made a pangenome of 18 elite cultivars plus the Chinese spring. That was the International Consortium main reference. We found 350 million bases that was additional to Chinese spring. And what we saw was that Chinese spring is very different to the elite varieties that are grown in Europe, the US, and Australia today. We found 245 genes in Chinese spring, which are absent from all the 18 cultivars. But there are over 12,000 genes that are identified in all the 18 cultivars. All these genes were found in all the 18 cultivars, but are not found in Chinese spring. And that's really quite important because if you're using Chinese spring as a reference to identify variation, identify genes that might be responsible for your trait and in your elite lines, there's a reasonable chance that the gene might not be in it because it's not in Chinese spring. So if you're doing any work on wheat, there are many better references now than the Chinese spring reference, and the pangenome reference is probably the way to go. This is a graph, a tree based on presence-absence variation, just showing you how far away Chinese spring is from all these mostly Australian lines. Since then, we've actually been working with the graph pangenomes, and within the graph pangenomes, we've developed a method to develop these. And we've adopted the Panache viewer, which was developed by a group in France. They used it to display the banana pangenome that we worked with them on. And the first wheat graph pangenome is available in this database. That's available publicly as a linearized form showing presence-absence variation of genes. Also, the data is available in draft form as well, which allows people to map their own data onto it to extend the presence-absence variation. And this is really the best way that we've seen at the moment to be able to generate high-quality graph pangenomes and to look at variations within graphs. And we're making these graph pangenomes for a range of different species now. While we were making all these pangenomes, I was approached by a friend, Henry Nguyen, who's based in the US. He's been working on soybean. Soybean is not a big crop here in Australia, but he said he had all this data. He had data from over 1,000 individuals from the US National Collection. And these include wild, landrace, old, and new cultivars. So we asked whether we could make a pangenome for these and have a look. And I thought, well, it's a beautiful piece of data. So let's have a play. Let's see what we can do. But first of all, he had to get the data to me. You can't just send this as an email. You can't even download it through the internet. It's the amount of data is vast. So I ended up receiving a large box full of large hard drives. We drove that over to the supercomputing center, just the other side of where I work in the other side of the river from where I work in Perth. And it took over a week to download that data onto the supercomputer. There was a crazy amount of data that was there. But we made the pangenome and we looked at presence absence variation. And one of the great things about having such a large and complex data set is you can ask really interesting questions. So for example, we can look at the frequency of genes in that population and compare the frequency of genes, for example, in wild compared to land races, or land races compared to modern cultivars. And these are just some examples. Here, each dot is a gene. They're colored by statistical significance. And as you can see, there are many genes that are showing high frequency in the wild lines. And when you actually look at their frequency in the land races, they're almost disappeared. They've been bred out. And again, in the opposite case, there are some genes that have got a relatively low frequency in the wild, but very high frequency in land races. So these genes that have been selected for during domestication. Again, to a lesser extent, you've got the early land races and modern cultivars. You're seeing changes in gene frequency between these two data sets. And this is a sign of selection for changes in gene content. And when you actually look into these genes and see what these genes are, it's quite clear why they've been selected for. You even see, if you actually look in greater detail, there's the US breeding, it's separated into the northern and southern breeding regions. And you're even seeing selection for specific types of genes between those two breeding programs, presumably associated with adaptation. And those sort of adaptations are going to be quite useful as we start to breed for climate change and changes in where people are growing these crops. One of the things we didn't expect to see, we saw lots of things we didn't expect to see, but one of the things we didn't expect to see was a reduction in gene content as domestication and breeding occurred. So if you look here at these violin plots, these are the number of genes in the wild population. This is the distribution. Some individuals have got many genes, some relatively low, but they mostly kind of sit around this area here. By the time you get to land races, the average number of genes has decreased significantly. So there's been a significant gene loss as you go from wild to land races. Older cultivars are quite similar to land races, not a significant difference. But when you get to the more modern cultivars, ones that have had more applied molecular breeding applied to them, again, you're seeing a significant drop again in these lines. Now, what's interesting about this is that this is clearly happening due to breeding and selection. There's a selection against the presence of certain genes. But breeders can only select against these when they have presence-absence variation, because otherwise there's nothing to select for or against. So there must be many genes which are potentially having a deleterious effect and breeders might want to get rid of, which are not showing presence-absence variation, and which could therefore be deleted, for example, using CRISPR or some other genome editing technology, which could again increase yield. And the identification of those genes that might have a negative impact is going to be quite difficult if they aren't showing very much variation. But there could be really interesting targets if we knew a little bit more about them. So where are we with graph pangenomes? Well, I think we're still very much at the start. We need to be able to build more and better graphs. Certainly graph pangenomes find missing heritability. That's very clear because you are actually capturing much more of the genomic information. The ability to put the information into formats that can be readily accessed computationally is still a challenge. And then when you want to start to overlay additional information, such as transposable elements, long non-coding regions, various chromatin types, genes, and allelic variation, when you start to actually put these into a format that can be computationally accessible and used in mining, I think that's really where we need to be. But I still think we're a few years away from that. So if anyone's working on this, let me know. I'd be really interested to hearing from you. So genomics, we've got our pangenomes, we've got all that genome variation. We need to link it to phenotypes. Phenotyping has traditionally been relatively manual. It's been challenging, but it has undergone a revolution. So we're looking now at next generation phenotyping, people using satellites, people using drones, people driving robots through fields. The technology is just taking off hugely. But it creates a challenge. You're generating a vast amount of data. So you've got all this genomic data, all this variation, and then you've got images and all these other high throughput tabular data. We can't just put that through standard statistical models. It just falls over. It's not really built for that sort of diversity. And because of that, we need machine learning. I'm not going to give a class here on machine learning. I'm going to give an overview of the sort of things that we use it for. Generally, you can take things like image data. So this is crop image data from fields taken with a drone. You can take text data, whether it's genome sequences or variation data. And you can take tabular data, whether it's yield or some other form of traits that's kept in tables. You put it through a whole range of different machine learning methods, and you can build models for classification. Is it this or is it that? You can build regression type models. So you can start to say, well, what's a yield likely to be? Can you kind of predict what that's going to be? Or you can cluster them into different types. And I'm going to give some examples of how we've used that data. So you take genomic variation, you take phenotype data, you can actually split it into test and train, build your models, then go through with the validation to get your final model. And this is an ongoing process. The types of models that are out there are changing continuously. So if you've got a data set that you want to apply these sort of techniques to, go out and have a look to see what the latest models are, see what's out there, because it's very difficult to choose the right model. You've actually got to go out and test quite a few. There's various data modalities. You can actually use multimodal deep learning, and we've been using this a lot more recently, where you take the different data types, whether it's genomic variation, an intermediate phenotype, maybe some environmental data, and some more advanced phenotypic data, and you build individual models for these. And then you actually bring these models together to make your prediction. This is really quite useful because if you have additional data coming in or additional inputs, or if you want to modify and fine tune some of these individual models, it's a lot easier rather than trying to build one massive model and then trying to rebuild it. So this is a really useful technique if you're using machine learning, is to take this approach. I'm going to give some examples of how we've used it successfully. I should point out that not all the data approaches that we've tried have been successful. There have been a few that were found really challenging and difficult to build models. We're still not sure 100% why that is, but we find that 70–80% of the data sets we've looked at, we've been able to build successful models for. This is an example of yield prediction in maize. So we're looking at images. These are various spectra from a drone image. And various manipulations have happened to these images to be able to make them suitable. One of the biggest challenges of applying machine learning into and building these models is to put the data in a format that is suitable for the model. And there's lots of different ways of doing that. And there isn't always a right or a wrong way. There's just different ways you can play with it. So we extracted the plots, normalized, we did some rotation, and we did a bit of standard machine learning tools where you modify the image slightly and then present it in a different way. They think it's just a new image, and build almost semi-artificial data sets from that. It's a standard way that's used in method that's used in image recognition. If we look at a table, we look at it from different angles and still recognize it's a table, the model needs to be able to do something similar. So within the model, we had just basic information on the parental lines, the seed stock. In one case, there's a fertilizer application, a bit of information on the planting date. And we're looking to predict yield from this data. I should point out that all this work is published, and if you've got any other questions, then feel happy to contact me. So there were 3 years that the images were collected and yield was collected, and what we found was a reasonable correlation between the predicted yield in the models and the actual yield. Now, it's not always a perfect correlation because these plants were imaged at 60 days post planting, so the plants are still very young. And there's no information on this model as to what is going to happen in terms of the environment. It doesn't know whether it's going to rain later on, or whether it's going to be a storm and wipe everything out. So that's why some of the correlations, these lines are not always perfect because it doesn't have environmental data. You can see the lower white and the upper whites. These are two different fertilizer treatments. It's separated these out pretty well. Now, one of the things that is useful to breeders is saying, okay, I'm not that interested in the final yield overall. I just want to know what the best lines are. And if we build a classifier model, it clearly shows that we can identify, even at a very early stage, high-yielding lines, and we're not getting low-performing lines being called high-performing lines. It's a pretty good model for making that distinction. So although because we don't have the weather information, we can't get the yield prediction perfect because it can't see the future, we can actually make predictions as to which one's likely to be the higher performer. We're building new models using similar data now, but including environment data, and we're looking forward to see how they work out. We had a vast amount of data from the soybean resequencing, so a lot of phenotypic data with that. We had quantitative and qualitative traits, and we had high accuracy in predicting these. And we showed that machine learning outperformed deep learning. We think that the deep learning models, there just wasn't enough data there. Even though you've got almost 1,000 individuals going into the model, we still think that the deep learning models, there wasn't enough data to be able to make the predictions. Machine learning performed very well. And many of the genomic loci, when you use explainable AI to ask what the machine learning model is using, which SNPs are considered important, those SNPs overlapped with the GWAS. So that gives us a reasonable level of confidence that what we're seeing is real. We did some R gene prediction in canola, and this was using phenotype assay, cotyledon score. Some of these resistance genes, the presence of some of these resistance genes is very difficult to predict because they're often masking other resistance genes. The presence of one can mask another one, but we've still got very high predictions for these. And this is allowing the automation of this type of classification of R gene content in canola. And this is built using an XGBoost model. It's one of the standard models that we know seems to work very well for this type of data. We looked at quantitative disease resistance in canola. So blackleg is a disease in Europe and Canada and Australia and a few other places that says a significant problem in canola. We've identified many genes for qualitative resistance, but we were interested in quantitative resistance, the minor gene input. And so we had multi-location, multi-year phenotypes. We had quite a lot of data here for this. This has not been published. This is proprietary data. We're not allowed to publish this, but I am allowed to show you this figure, which basically shows that we've got a reasonable correlation based on SNPs. We can take the genotype of the canola and predict survival percentage in the canola blackleg trials. Again, we thought this was going to be more of a challenge. In fact, it still was a challenge, but we got better results than we expected for what is something that's really quite difficult to predict, which is quantitative resistance. I would love to play with other data sets with this. If anyone else has got similar data sets and wants to work with us, send me an email. Again, would love to work with you on this space. So where are we going next? So we need more and better annotated pangenome graphs. We're still in the early stages of pangenome graphs. There's still a lot that we can do. There's a lot more technology that's needed in terms of being able to build better graphs, build graphs that can be accessed computationally more efficiently, and also to be able to host a greater variety of information. And we need more and better machine learning and deep learning models for prediction. Again, we're still very much in the early days playing around with this. We're scratching around for whatever data we can beg, borrow, or steal to be able to build these models, because it's data hungry. We need more data. And we're looking to put as many diverse data types in as possible and seeing how we can transfer data from one type of model to another type of model. There's a huge amount of that could be done in this space. What's really useful is we can reuse old data and add this data to new models. And then we need to optimize the path to breeding improved crops. And this involves working very closely with breeding companies. And we've got a few funding applications at the moment with some breeding companies to allow us to build a path to help improve crops using these methods. So we need bioinformatics. We need bioinformatics to support and accelerate breeding. Is there a flood of data? Certainly there is, but we need more. There's a lot more data that's out there that we need to be able to get. There's some lovely tools that are out there to be able to build all these lovely models and put these data into places where it's usable and accessible. We need a lot more, and that's what's driving me to develop the Master of Bioinformatics. We're developing micro-credentials in bioinformatics, and we're telling everyone how wonderful bioinformatics is. It's a fantastic job. You can sit here on the coast and just enjoy lots of lovely emails and work that way. It's very flexible. But where's the future? This is more challenging. We are continuing to burn fossil fuels. The speed of climate change is unprecedented, and we are seeing this. When I first started teaching on the area of climate change in agriculture, some people were still denying. No one's denying it, where everyone can see the evidence is out there. The impact is unpredictable, and we may not see the tipping point. So I'm going to leave it up to C3PO to bring the final conclusion into that. I should point out that I didn't do any of this work. I have a wonderful team, past and present, and some amazing collaborators, many of whom you'll see on the publications. And people still give me money, which I'm always very grateful for. And with that, I'm very happy to pass back to Anna and answer any questions. Thank you very much.
Thank you very much for such an interesting talk. And as you can see from the questions, a very inspiring and thought-provoking talk indeed. So we do have quite a few questions in just a few minutes to run through a few of those. So I'm going to pick a few out. I think one of the main threads that came from the questions is an interest in the kind of software and tools that you've been using for various stages of the of what you talked about. So the first question I have, what kind of software do you use for building the pangenome, mapping the reads, and also calling SNPs? It's a few questions in one there, but for the different stages.
So we're quite conservative in the mapping. So there's a range of different software that you can use to map reads. Some of them tend to, BWA tend to overmap. They tend to force reads into position, and we don't really want that if we're identifying variation. So we use Bowtie 2 for that sort of mapping. For graph pangenomes, we put things into giraffe format, which seems to work quite nicely for read mapping. Most of the methods are on GitHub and also in the publications. So Bowtie 2, I think, is one of the main things that we use for mapping. For SNP calling, BWA works fine if you're just using it for SNP calling, where you're likely to identify more SNPs because it will actually map more reads. But as you change the process of read mapping and make it easier to map the reads, then you're going to get more falsely called SNPs as well. So it's a balance. It's always a balance as to what you're going to use the data for in the end.
And there's also an interest in how you have created the shack plots that you showed earlier on.
Well, the shack plots are really cool. And I'd have to pass that on to one of my team who actually makes them, because I don't physically make them myself. Whoever asked that question, send me an email and I'll put you in touch with the person who developed that. But they're really nice. We're actually writing a paper at the moment on explainable AI and different ways that it can be applied. And I'm also happy to share a preprint of that as well. If it might not even be preprint really, I'll share a draft version if people are happy with that, and promise not to share it.
That's great, thank you. And just to mention, we will share the slides along with the recording, so you'll have the email there to contact. And another question around software and tools was, I think you mentioned a machine learning method that you used, but what kind of software do you use to do your machine learning, particularly for image analysis is a question.
Oh, for image analysis, I'd have to go back and look at the methods, because we try pretty much everything. The challenge with machine learning, it's you've got the no free lunch hypothesis, which some of you may have heard of, which basically says with whichever data set that you're planning to use, it's impossible to predict which method would be the most accurate. So basically, you've got to go out and try everything. XGBoost works very well with tabular data. We're very fortunate to work and publish and get funding with the collaborator Mohammed Ben-Amoun, who's a machine learning vision expert here at UWA. So he'll often send me an email saying, we've got this new method for image analysis, go and try it out on your crop data. He does it for factories and going around and identifying objects in factories, that kind of thing. But I persuaded him that crops are useful. So it's an impossible question to answer, because there are so many different methods. And even when you look at the methods that were published, by the time you get around to publishing, a lot of them are out of date. So you've got to keep on keep on looking to see what's out there. Transformers are really quite popular at the moment, and that's the basis for the chat algorithm that gives you answers to all your student questions. But there's lots of new things coming through all the time. Find a friendly AI expert.
Yes, indeed. So moving on from the software side of things onto the actual data itself, someone asks, are there any reference pangenomes available for download and use for one's own genomic analysis?
There are many. So everything that we've done pretty much is published and available. So all the wheat and the Brassica ones are there. There are several soybean pangenomes you can download and use now, of variable quality. I guess the thing to think about though is that they're changing all the time. So the Brassica pangenomes that were published, they're now outdated. They were published a few years back, and we're making graph pangenomes using the same method that we used for the wheat graph pangenome. So I wouldn't use our very first wheat graph pangenome data. I'd use the latest one. It's built with much better data and much better models. In 3 or 4 years' time, we're likely to be making them again as the technology changes again. It's a rapidly moving field, but certainly there are plenty. And if there's a particular species they're interested in, if they want to get in touch, then I'll let them know if I'm aware of what's out there. But for most major crops now, that there are pangenomes, and even quite a few developing crops as well, there are pangenomes. It's relatively cheap now to actually go out and generate the data. If you want to go out and sequence 10, 20 individuals and generate whole telomere to telomere assemblies, as long as it's not something as horrible as wheat or Paris japonica or something bizarre with a weird genome, then it's relatively easy to put together.
Okay, thank you. So we're coming up to the hour here, so I think we'll leave the questions there for now. But obviously, as we've mentioned, we'll share the slides, and that has a contact email. So don't feel like you can never ask a question again. This wasn't your only chance.
Yeah, and my email address is on the slides, and feel free to contact me. I'm really keen to work with people and to build collaborations. That's how we get publications.